Right.
Başladı.
Dr. Ayar had his undergrad education in veterinary medicine in Ankara University.
Bunu
And
görüyor mu
then
acaba?
he
Yok
had
sen tamam başladı.
PhD in London and Scotland. About the universities, he is a physiologist, he's a neurophysiologist currently working in Karadeniz Technical University. He's going to talk about the calcium imaging in neurons. Yes, the screen is yours. Ahmet.
And everybody,
You may like to.
okay, thank you, Chairman. You just made it Scotland, but last time we were there, you took several photos. You still have to show me the photos last time you took in Scotland.
Especially the sea seals, okay?
I'm looking forward to it. So, I'm going to talk about fluorescent calcium imaging. I really enjoyed the talks before me, and I realized some of my slides are a repeat of Dr. Emre's presentation. So, in the meantime, I tried to change a little bit a focus of my talk. So what I'm going to do, I'm going to focus on technical issues, and I will try to raise questions, if not answers, for those who are interested in buying a calcium imaging system or deciding which system best for them. So I will try not to repeat the detailed explanation, especially by Dr. Emre. So, why study calcium? Why do we have to bother about measuring calcium in cells? Because calcium carries messages, virtually all important functions, not only physiologically, but also in pathological conditions. Calcium is a diverse second messenger, which involved in the muscle contraction, cell motility, phagocytosis, cell division, fertilization. The life starts with calcium and ends with the calcium, large calcium changes in the cellular level. It is involved in neuronal signaling, and maybe the most relevant part to this meeting is secretion, neuroendocrine and endocrine secretions. Furthermore, calcium levels or changes in free calcium levels inside the cell is the final stage of cell death and injuries in hypertension, in cardiac arrhythmias, in some hematological problems, muscular diseases, pain disorders, which I have interest in peripheral mechanisms of pain, and in many psychiatric syndromes. If you are, I'm trying to give a message to the young people who are attending this workshop. If you are interested in performing research in any of these areas, whether you are interested in investigating physiological mechanisms or try to contribute to pathophysiology of any of these diseases, or investigating potential new drug molecules, use of calcium imaging or measuring intracellular free calcium going to give you power to answer your research question. As I said, I don't want to repeat the previous talk. I will add up, and you may have to combine my presentation with especially Dr. Emre Yasin's presentation. So we are interested in measuring, following cytosolic free calcium levels. So we have to have a good knowledge of calcium handling or calcium homeostasis of the cell. Calcium is present virtually in 4 compartments. It's large concentration at millimolar levels at the extracellular space, and about 10,000 times less in the cytosol, which is in the range of 0.1 micromolar. And after stimulation, it never gets equal with the extracellular space. So it increases, but still far below the extracellular space. Calcium is also present in intracellular calcium stores. These so-called stores are endoplasmic or sarcoplasmic reticulum. Endoplasmic reticulum contains the second largest concentration of calcium, which is sequestered in these cellular organelles, and in appropriate signal, these intracellular stores release calcium and contribute the increase in intracellular free calcium levels. And the free calcium level is the key to induce hormone secretion, contraction, gene expression, and other physiological and pathophysiological conditions like apoptosis. The mitochondria also contains as a store calcium. The concentration in the mitochondria is in the middle. It's not as large as endoplasmic reticulum, but much higher than the cytosolic level. When we interest in mechanism of a physiological process or a pathology, and if calcium is involved in this process, we have to have a view of what sources the calcium is increasing, what sources the calcium is arriving from. Calcium as an ion, although very large at the extracellular space, cannot enter inside the cell. So increase in free intracellular calcium can be achieved by passive diffusion, mean moving from higher concentration to lower concentration. But the cell membrane does not allow calcium to enter inside the cell freely. So there are channels over the membrane which allow, under strict control conditions, the entry and a signaling of calcium. These are involved voltage-gated calcium channels, which are many subtypes according to their biophysical and pharmacological properties. TRPC channels, so-called transient receptor potential channels, they mediate calcium entry under physiologic and pathophysiologic conditions. TRPC channels, NMDA channels. They are classical receptors for excitotoxic or excitatory neurotransmitter, AMPA, kainate, and related neurotransmitters. So through these channels, calcium can enter. There is a recent discovery about another class of calcium entry route which are related with the intracellular stores, yet undefined factor sends signals to the membrane when the intracellular calcium stores are low or depleted, and they stimulate entry of calcium through aura channels. The intracellular stores can further release calcium to contribute to calcium-mediated signaling. There are two main types of calcium release channels over the intracellular stores. One is sensitive to IP3. The IP3 sensitive calcium release channels also sensitive to caffeine, cyclic ADP ribose, sphingolipids, and others. There is another type of calcium release channels present over the endoplasmic reticulum, which are the ryanodine receptors. Calcium itself, once entered inside the cell from the extracellular space, can cause release of further calcium from intracellular store, which is called calcium-induced calcium release. So these mechanisms, alone or in combination, can cause increase in calcium and thereby activate calcium-sensitive elements and modulate the activity of certain enzymes, certain proteins to induce physiologic or pathophysiologic processes. Cell has to handle calcium very tightly, should not allow increase in intracellular calcium, so it has mechanisms to rapidly and efficiently remove calcium from intracellular stores. The calcium is taken back in one mechanism to the intracellular stores by Cerca pumps, which they must use ATP. There is also PMCA, plasma membrane calcium active pumps, which has to use ATP. Sodium calcium exchange pump, I have not included, it's not included here, also help to remove excess calcium and maintain the resting level after calcium being involved in signaling activity. The ATP cost is huge, but it deserves it, because if calcium levels remain high, it will induce apoptosis and destroy the cell eventually. This slide summarizes what just I talked about, the mechanisms through which intracellular free calcium increases, which involves phospholipase C activation, producing IP3 and diacylglycerol, which also has a stimulating effect on TRPC channels, further causing increase in intracellular calcium. The free intracellular calcium can bind the calcium binding proteins, as well as other targets like calmodulin and other cellular targets to induce calcium-mediated activities. By summarizing, the role of calcium in physiological and pathological processes, we answer the question why one needs to measure, why one needs to deal calcium. Calcium changes in cells, including neuronal and endocrine cells. But it's worth measuring. It will be important. It will provide information. But the second question comes, how can we measure? So we have to know about some requirements and some challenges. The calcium itself cannot be visualized directly. Okay, as Professor Alcan indicated, we have strong tools. Biotechnological support has increased the capacity of microscopes, but we cannot directly visualize calcium. And the mentioned calcium signals are very fast. So you have to have a very strong and timely, efficient system to detect changes in calcium. And we also need to measure calcium dynamics in living cells. So these are the requirements. If we look at, I'm trying to make a story to those who are new in the field. Okay, you need to measure, you're now persuaded you need to measure calcium, but you don't know how or which technique to use. If you are going to buy, which technique you should invest. Emre emphasized electrophysiologies still are popular. I spend a lot of time performing patch clamping. Although it's a headache, I still miss it. It's a strong technique, but it has some limitations. You are measuring the electricity, you are measuring the current. So you cannot easily differentiate between cations, whether the depolarization is coming from calcium or sodium entry or exit of potassium from the cells. Of course, there are approaches and protocols, you can examine them, but it's indirect way or additional experiments. Calcium selective electrodes came into use, and vibrating calcium selective probes, which I'm not going to focus on because I will talk about fluorescent calcium imaging. Digitized video microscopy now enhanced our ability to follow calcium, and we have variety of options. We can use confocal laser scanning microscopy, 2-photon exciting laser scanning microscopy, and if you are interested in what these do, you probably have to get in contact with Professor Gürkan because they have an amination of these equipment in Istanbul. Flow cytometry, photomultiplier tubes are other techniques. Is it a matter of finance? Is it a matter of money? If you have enough money, should you necessarily go for confocal laser scanning microscopy? If you cannot afford this, or do you really need this? So you have to question, what are the technical challenges for calcium imaging? First, you need a level of contrast, specifically for calcium. You need to make quantification. You need to measure how much calcium has entered and is it a global or is it in subcellular area? Because sometimes you are interested in only certain parts of the cell and where you expect some interactions. Since the signals very high, very fast signals, but we should not generalize all calcium signals are fast. Depending on the preparation, the cell or the tissue sample slice, and nowadays in vivo calcium imaging is available. You need to know the nature of your calcium signaling in those tissue. This is one aspect of whether you need an advanced, expensive tool or a cheaper one would be okay for you. Of course, we also need to consider toxicity, pH issues, and bleaching of the dye we are going to use. So spatial and temporal precision is one requirement. Now it's over 30 years calcium imaging, fluorescent calcium imaging in application, and nowadays we do have a high temporal and spatial resolution techniques. And now, using these tools, we can concentrate on subcellular and tissue levels. And newly, Professor Bayram Yılmaz bought for Turkey, maybe the first system, in vivo calcium imaging system, which also allows you a high temporal and spatial resolution with advanced technology. Anyway, you may not be lucky to work in one of the private universities in Turkey, and you're still interested in using calcium imaging techniques that you can afford, or you don't want to waste your money, which is one of the main problems in Turkey. People buy jet planes and just go to work. I mean, they can easily do it with their car. It's a waste of money in my regard. So you have to ask yourself, okay, which signal you are going to follow? Is it going to be extremely fast? Mr. Chairman, can you hear me? Is everything okay?
Yes, everything is okay. Please go on. Yeah.
Because I had a problem on my screen. I don't want to talk on my own.
Yeah, just
Thank you. Is it ultra fast, or are you interested in following the signal over hours and days? Of course, they will require different capacity of equipment, including the cameras and PC system as well. So you have to know your signal. How large is it? How fast is it? I'm not going to talk about, because I don't know whether your interest is, but I will give you a clue. Calcium signals can be very fast and big in amplitude. These are a potentiation of synaptic efficiency. calcium signals can be long-lasting. I mean, you have to have a time resolution suitable for this. But for this, the time resolution is not the main issue, the specific sensitivity issue, because the signal level is low. And some calcium, some tissue, or in some mechanism, calcium signals can be in between these. And you have to know the nature of your cell or tissue in terms of calcium signal. I mean, you have to, if you measure this type of signal and measure it here, you miss the real peak. And if you are going to apply an agent to block this calcium, it's already tiny. If you investigate it over here, you miss the first transient rise. If you miss this, you are in trouble. I mean, you are technically in difficult position. So it's not only issue of technology. We need to concentrate on what signal we are investigating, focusing, and what is the normal nature and type of the signal. Emre has clearly explained the basic mechanisms of fluorescent excitation and emission mechanisms. I'm going to talk about the basics of equipment required. Of course, we need a microscope. And I have to thank again Professor Altcan, because I had attended his workshop years ago. He talked about the importance of objectives. We need a light source, we may need filters, need camera, and operating system and software. Of course, fluorescent labeling and perfusion system. Sorry, this is in Turkish. So,
Dr. Ayar, can you a little bit speed up?
Okay, I will. I can stop anywhere else. I mean, how long do I have? How many minutes?
Actually none, but just I'll give you 5 minutes, okay?
Okay, in 5 minutes I will stop.
Okay.
So what you do, in case of using isolated cells, you can place your cells over the cover slips, and you can perfuse them through a perfusion system. There are automated systems, or you can just manually open one, give them a wash, and remove the perfusion system. So the objective is attached, and the CCD camera receives the signals, transferred to the computer, and we digitize and measure. I will just make an emphasize, the microscope is one of the key parts, but we have to concentrate on what objective is, because the working distance and numeric apertures is critical for this application. And the price of objectives change maybe 10 times. I mean, you can get a decent objective for €1,000, but for high efficient objective, you have to maybe pay €15,000. So the choices of microscope is an issue, but maybe the objective is the key, one of them. So for the dye you select, you have option of different light sources. So you have to decide again which light source is useful for you. I mean, mercury has certain limitations. Xenon has some advantages. it also needs to be considered as one of the parameters when you are deciding to which light source to use. And the CCD cameras, they are another critical part of the system. Their speed, resolution, readout time, they make huge difference for the price. And which one you are going to use also depends on the nature of the signal you are going to follow. I'm not going to talk about for the dyes. You can always read detailed reviews about that. Fura 2 is a ratiometric dye we use, and once you detect an increase in free calcium, you also check the bind calcium. That's why it's preferred for some applications. The dye loading protocols is also important because if your system is not robust enough, you may need the acetoxymethyl ester may not be useful for you. Using more invasive techniques like microinjection, you may end up losing time, durability of the cells. So basically, I will show you one more original slide from my studies. We do primarily isolate sensory neurons. We load them Fura 2, and we have a pseudo color or black-white images. And using software, we can measure intensity of calcium signaling. To conclude, fluorescent calcium imaging is a strong tool. We can target specific cellular and subcellular level. Its productivity is high. You can measure signals from many cells in the field of view. The cameras are useful for this purpose. I would like to just thank some colleagues. They helped me in establishing fluorescent calcium imaging in 2 different universities in Turkey. And sorry for the time, Mr. Chairman. If we have a question, I can take some questions.
Yeah, thank you very much, Professor Ayar. Maybe anyone who likes to ask questions may directly get into contact with you and get some practical advices. I need to switch to the last presentation by Zeiss. Ahmet Ayar is going to speak about artificial, let me exactly, mathematical modeling in, sorry, right. Let me find artificial intelligence sample finder. I'm mostly aware of Zeiss technologies, but this is the first time I heard about this title. And after all these praises to advanced microscopy, I hope Erma will announce that attendees to the 50% discount, of course, on their first. Of course. Okay.
Thank you. My name is Ahmet Ayar. I am working for Zeiss Microscopy Department for more than 8 years. And thanks for Professor Ahmet for